I'm on TikTok. It's really great. So Helena got her BS in biological sciences in Wellesley College, and then worked for several years at the Woods Hole Oceanographic Institute, which incidentally where we first met when I was visiting. I brought 2 graduate students with me on that visit. And then joined SAFT in autumn of 2019, first as a master's student, and then bypassed into the PhD program shortly thereafter. In her time here, there's a bunch of things I wanted to say. I'm realizing I wrote way too much, but sort of 2, I guess, main themes I wanted to talk about is just how much of a leader she's been, how much she's sort of given back to the SAFT community. That's sort of taken the place of a bunch of official roles, so like various committees, like search committees. There's the GPSS, to get that acronym right. She served as the GPSS, like the Student Center, is that the right word? And probably some other things I'm forgetting about. She's done a lot of like teaching, just, not just because like I need to, but it's like, hey, I want to do this. I want to learn about teaching and I want to get back. So that's like a bunch of official roles, but like she's also unofficially just been such a important contributor to the school. I know I just routinely sort of found myself consulting with Helena, whether it was like issues within our lab we wanted to talk about, like policies or departmental policies. It's always just a really trusted advisor, and I always got great guidance. So it's really remarkable. And then also been at the same time a really productive scholar as well. You're going to see a taste of what she's been working on, but her interests are pretty broad, and she has 9 published papers, so she's an author or co-author, including 2 of her dissertation chapters, which is amazing. And then just generally, I wanted to note just about her scholarship in general. Every person has like something that's really noteworthy, and for me, the thing that really stands out about Helena's scholarship is she came here, had her working at UI, and kind of had a foundational interest in sort of biological oceanography. And what I find really remarkable and great about Helena's work, and you'll see evidence of that soon, is she's really held on to that sort of core interest of like, hey, I'm interested in sort of questions that sort of in the broad realm of biological oceanography, but I'm here at SAFS, and then I'm learning how people in SAFS sort of think about problems, how they answer problems, and she's done this great job of kind of melding those 2 fields, both in terms of methods and frameworks and approaches to asking and answering questions. And I've learned a great deal on the biological oceanography side from working with Helena. And I hope she's learned a little bit from me on the fishery side. So with that, please join me in welcoming Helena McMonagle.
Okay, thank you so much, Tim, for that introduction. That was really generous. And thank you everyone here for tuning in online and for coming in person. It really it means a lot to me, and I'll miss this department and you all a lot after I head out of here. So I'm here to tell you about my dissertation, and I'm going to start with talking about these beautiful fish that I've been studying for the past 5.5 years. That we call mesopelagic fish, or twilight zone fish. And these are some of the families. There's the topids, hatchetfish, bristlemouths, and these are the most abundant fish that you've probably never heard of. I hadn't before starting on this research journey. But there's been a recent discovery that these fish are about 10 times more abundant than we previously thought. And this discovery has sparked interest both from the commercial fishing industry and also from a carbon cycling research community in understanding the role that they play in carbon cycling. So there's sort of this trade-off between potentially using this new food source versus better understanding their role in how they move carbon through the ocean, which connects to climate change mitigation. All right, so there's been a fair amount of media on this topic around the time that I was getting interested in the role that fish play in carbon cycling. And this is an article that came out in the New York Public Radio, again, trying to understand and maybe expressing a bit of concern about trade-offs between carbon cycling versus commercial fishing for these species. This is an article in a sustainable development think tank on this topic. The US Navy has published on this topic. Oceans Deeply is Ocean Magazine, Kai Magazine, NPR. But it was this Guardian article that really, really kind of caught my attention when I was starting off in this research topic. And it was titled, "In the Ocean's Twilight Zone, a fish that could feed the world or destroy it." And this kind of language sort of made me want to take a step back and look at what do we know from a scientific perspective about this, these concerns, these trade-offs between commercial fishing and other services these fish provide. And so I looked back into what we know among the scientific community about this topic. All right, so we know that the oceans absorb about 1/4 of our carbon dioxide emissions. And this happens through a variety of different ways. And it turns out it's really important to understand how much carbon is coming into the ocean from the atmosphere in the form of carbon dioxide, and how much is the ocean off-gassing if we're going to predict the future of climate change. There's two kind of main categories of ways that the ocean absorbs this 25% of our emissions. The first is the solubility pump, and this is all the chemical and physical processes by which the ocean absorbs, transports down deep, and stores some amount of carbon dioxide from the atmosphere. There's also this biological component to that, and all those physical and chemical processes aren't enough to explain this 25% that the ocean absorbs of our emissions, and so the rest is thanks to life in the ocean. And this ranges from phytoplankton that grow and incorporate carbon dioxide into their tissues, like trees do, for example, to larger animals like fish or zooplankton that transport carbon down deep. And something to know also about the carbon cycle is that the deeper carbon gets stored or transported into the ocean, generally the longer it's going to be sequestered from the atmosphere. Okay, so that also matters if we're going to talk about climate change. It's not just carbon transport, but carbon sequestration on long time scales. All right, so going back to carbon transport, here's what we know about the biological carbon pump and carbon transport. Anywhere between 5 and 12 billion tons per year of carbon is transported from the sunlit surface zone to the twilight zone. That's a really big range of uncertainty. Humans emit about 10 billion tons every year, just to give you a sense for how uncertain we are about how much biology transports carbon into this layer of the deep sea. Another mystery about the biological carbon pump is that when we go to make a carbon budget for the ocean, the carbon accounting doesn't quite add up. So what I mean by this is that we measure more carbon in the twilight zone than what we're able to account for by all the different pathways that carbon gets down there. Okay, so there's some kind of missing sink that we're not considering. One of those potential missing sinks is the role that fish and zooplankton play in moving carbon down deep. So it turns out fish in particular aren't even included yet in these big global models of carbon cycling, which are what the UN and the IPCC use to predict climate change. And so, little sunrise there. Lights just came right here. So I was really interested in whether these twilight zone fish could be part of the answer to this sort of mismatch in carbon accounting in the ocean. All right, why else did I focus on these twilight zone fish? As we learned before, there's about 10 times more of them, we think, than we previously thought. These biomass estimates, though, were really uncertain. We used to think there were about 1 billion metric tons of these fish. Now we think that could be as high as 10 or even 16 billion tons. Just for some perspective, that means that these fish could be making up anywhere from 50 to about 95% of fish on the planet in the oceans. And that also means that if, say, there's 10 billion tons of twilight zone fish, that means there's about 100 times more of these fish in the ocean than all the fish that we harvest every year. Okay, so that's one reason to focus on them. Another, like I said, is that they may be commercially harvested soon, and they would be the proposal so far to use them for nutritional supplements and also for fish meal to feed our livestock, our farmed fish, and potentially our pets as well. So there's interest in kind of using this as a way to meet a growing human demand for food. And I also got really interested in this part of the carbon cycle because it's a part that human action and governance can directly influence, right? We maybe can't control the waves or the ocean currents, but this is something that if it's important, our actions can make a difference. Okay, so I've talked about fish and moving carbon around. How does that actually happen? One of the major ways that these twilight zone fish move carbon from the sea surface down into the deep, where it can be stored for longer time scales, is through this process called vertical migration. So vertical migration means that twice a day, these animals are moving up and down from the sea surface down to like 500 meters in some parts of the world. And that's like running a marathon for a fish this big every day, up and down. And so what happens is these fish during the day, they're down in the twilight zone where they're hiding from visual predators. And then as the sun sets, they come up to the sea surface at night and they fill their bellies with as much food as they can eat, where there's a higher abundance of food at the sea surface. And then when the sun rises, they come back down into the twilight zone where they release some of that carbon that they've eaten in their food the night before. All right, so I wanted to then look at, all right, what do we know in the scientific community about the role specifically that fish play in carbon cycle? So let's imagine this blue line sort of represents the active carbon transport by fish. And this green line represents the sum of active plus passive carbon transport. Passive carbon transport is, for example, the growing, dying, and sinking of phytoplankton that sink down. What we see in this review paper that conveniently came out around the time I was starting grad school is that if you look across, there's not that many, but you look across the papers on fish-mediated carbon transport in the ocean and kind of synthesize that into this literature review, they found that fish could be contributing anywhere from only about 3% to the total biological carbon pump globally to maybe as much as 29%. Okay, so this is a huge uncertainty range in estimating the global contribution of fish. And so that's something that I wanted to dive into to figure out, well, what do we really know about how much carbon these fish transport, and how can we constrain that uncertainty further to get a more useful estimate than we have so far? So this is the overarching question of my dissertation. How much carbon do these fish transport and also sequester for long time scales? And how can we start to tackle uncertainty in these estimates? So yeah, I was interested in kind of testing this idea about whether these fish are the climate change superheroes that they're sometimes made out to be. All right, so in chapter 1, I kind of go back to basics and I figure out how much does an individual mesopelagic fish transport, and what are the main sources of uncertainty when you go to calculate that. We'll go into this in more detail later, but in chapter 2, we basically scale up to the ecosystem level. And in chapter 3, we try to look at whether we can use new measurements to actually constrain this uncertainty with new data. All right, so starting with chapter 1. The first step was to develop a model of fish carbon transport, and this is publicly accessible code. Anyone can build on this, critique it, improve it. And what this model looks like in kind of graphical form is that you have a migrating fish. It's up at the sea surface at night, comes down during the day into the twilight zone or the mesopelagic zone, and it releases some of that carbon that we talked about that they eat near the sea surface at night through its respiration. So these fish breathe out carbon dioxide like we do. Through its mortality, these fish can get eaten by things that remain down in the twilight zone. We allow that to vary because there are some surface level animals that eat them too. And then there's their waste products that also drop off carbon down deeper. And this includes their digestion, which is basically fish poop, and their excretion, which is basically fish pee. And then we also, in this model, have a component where we're estimating the carbon transported by a non-migrant fish. So I talked about vertical migration, but not all fish do that vertical migration. A lot of these twilight zone fish just stay down in the twilight zone. Okay, how do they contribute to carbon cycling then? Well, for one, they eat some amount of their diet is these vertically migrating zooplankton. And so when they eat those, they're preventing those zooplankton from coming back up to the sea surface the next day and potentially releasing more carbon up there. Okay. Next in the model, we have to choose the flux boundary. So that's the depth below which we count carbon as being transported. A lot of the research on the biological carbon pump has been looking at the carbon transported past 200 meters. That's the top of the twilight zone. We allow that to vary, but 200 meters is a pretty common flux boundary to choose for carbon transport. All right, so we've created this model. It turns out when you really systematically look at all the different pathways that a fish can transport carbon, there's about 10 or so equations that we use to calculate this and about 30 different parameters in those equations. And what I mean by parameter is it's, a part of the equation that you need to assign a value to ultimately calculate fish carbon transport. So we look in the literature and we figure out what do we know for each of those different parameters would be a reasonable minimum and maximum estimate. So what I mean by that is we need to know the digestion rate of the fish, for example, how much carbon is in those fecal pellets that they produce or their poop. How much are they respiring out carbon dioxide? What's their swim speed? How deep are they migrating, etc. All right, the next step was to do the sensitivity analysis. This is called a Monte Carlo simulation. And this is really cool because it starts to help us understand uncertainty. All right, so let's imagine for each of these different parameters I've mentioned, there another number in this set of 3 different numbers in a slot machine. So every time you pull that handle, you're going to get a new set of parameter values, right? Here it's 3 sevens. Next time you might get a 7 for parameter 1, a 6 for parameter 2, 2 for parameter 3, whatever. But we have a lot more than 3 parameters. So we basically have a gigantic slot machine. Do that, and with every pull, you get a new set of parameter values. And you get a new estimate of fish carbon transport. What we did next was we repeated this about 1,000 times. You can do 1,000, 5,000, whatever. So what that does is it gives you not a single point estimate, which is sometimes what is done more in the literature, but rather it gives you a distribution of all the uncertainty propagated across all this different parameter uncertainty when you go to estimate how much carbon an individual fish transports. So this is what that distribution looks like. Let's focus on the red for now. This is the carbon transported by a single migrating mesopelagic fish. So as you go farther along the x-axis, that means they're transporting, your estimate is predicting that they transport more carbon, and the y-axis is how common those estimates are in that range. So what we see is that there's quite a wide distribution of how much carbon a single fish transports. But if we look at the average value for migrators and for non-migrators, what we see is that migrating fish are transporting about twice as much for a fish the same size as a non-migrator. Ultimately, what we're going to want to do is figure out, well, okay, how many migrators and non-migrators are there so we can scale up, but that's in chapter 2. Next, like I said, this is a pretty broad distribution. It's not super tight for either of these fish, especially the migrators. And so it turns out there's about a 6-fold uncertainty from this 5th to 95th percentile in that distribution. That's a really big range, a 6-fold uncertainty range where these fish are transporting anywhere from 1/2 to 3 times their body weight each year. All right, the next cool thing that the Monte Carlo analysis allows us to do is to pinpoint which of these different parameters in that calculation are contributing most to that uncertainty. So if a parameter looks something like this, it's varying between 1 and 6, and depending on whether it's 1 or 5 or 6, there's quite a big difference in the carbon transport in the final calculation. So that would be a highly influential parameter. This would be a not so influential parameter where it doesn't really matter what parameter value you choose from what we know about this value in the literature. On the y-axis, you get about the same carbon transport either way. All right, so when we do that and we look at all these different parameters in the calculations, this is for migrating fish. What we see, which is good news, I guess, is that a lot of these slopes are pretty flat. So that means that doing further measurements on something like the carbon content per weight of fecal pellets, it's not going to get us that much closer to figuring out to constraining that uncertainty. But there's a few parameters that do have a really high slope, and these are, for example, some of these respiration rate parameters that I'll talk about more later, and also some of these digestion or fecal pellet related parameters, like this export ratio that I'll talk about in a second. All right, so these are the ones that we'd really want to pinpoint if we want to constrain uncertainty with new measurements. There's also, like I said, some not so influential parameters. For example, the excretion rates of these fish are not making a huge difference because it's quite a small component of carbon transfer. All right, so I mentioned that respiration rates are really influential in these calculations. What do I mean by that? So this is what we base our respiration rate on when we do this modeling. And this is from a paper that was published in 2016 by a Japanese scientist, Dr. Aikita. And what he did is he looked across all the papers that we have so far on where someone has actually measured mesopelagic fish respiration rate. And these are all the points shown in black. There's not that many. And so, yeah, on the x-axis is the mass of a given fish, and on the y-axis is the respiration rate for a fish of that size. So yeah, you get an estimate of respiration rate for a given fish size. But because this isn't a really tight relationship, we don't really know if this line should look more like this or more like this. There's some uncertainty there. Another important source of uncertainty is this activity scaling factor. So this is how much you scale up the respiration rate predicted here when the fish is more actively foraging or migrating, when it has a higher metabolic rate. And depending on what you choose for that activity scaling factor, you know, this line is anywhere from here to here, and that makes a big difference too when you go to calculate carbon transport. I mentioned that there's also some fecal pellet related parameters that are really important. And one of those is the fecal pellet export ratio, which is just what percentage of the fecal pellets that are produced are we going to assume sink below that or end up somehow below that flux boundary. So that's another important source of uncertainty. All right, so we've identified some places where future research could do kind of give us the most bang for our buck in constraining uncertainty. We know that there's about a six-fold uncertainty range for how much an individual fish transports when we consider all the uncertainty. And some of these respiration rate and ingestion rate parameters are some of the most influential when we go to look at how much carbon a single fish transports. But what we really want to do is figure out how much do fish transport at the ecosystem scale, right? How much does a whole population of fish transport? And what is the relative contribution of fish to this biological carbon pump? So in order to get at that relative contribution of fish, what you really want to do is have a super comprehensive sampling effort where you collect information about how much passive carbon transport there is by dying and sinking with phytoplankton, for example, and how much active carbon transport there is in the biological carbon pump through diavertical migration of fish and zooplankton being kind of the most abundant animals. That is not a small amount of sampling, and it's really rare to actually have a chance where you're directly measuring all of these different pathways of carbon transport at a given time and place. So we were really fortunate when the opportunity arose in 2021 in May, during the pandemic, where we got to go out on this 3-ship operation where we met in this really well-studied site in the North Atlantic off the coast of Europe. And this was a huge collaborative project funded by NASA and also Woods Hole Oceanographic Institution. And basically, I was on this ship, and we were studying fish and zooplankton. There was another ship studying zooplankton, and others were studying that passive carbon transport. So before we set out to sea, here we are putting together this net that we're going to use for catching fish and also zooplankton. And the net that we use is called a multiple opening and closing net and environmental sensing system, which is abbreviated to MOCNES because that's a really long name and it is kind of a monstrous net. And this is a really cool technology because what it does is you send this net down to say 1,000 meters, and then as that net comes back up through the ocean, you open and close these nets at different depths. And what that gives you is stratified samples that tell you how many fish and zooplankton are there at a given depth. And what we do is we tow this net during the day and night so that we can figure out how many of these animals are migrating and how many are staying down deep, and how deep do they migrate up and down. All right, so we ran into a bit of a problem while we were out there with this net, which was that we came across some of the worst sea states that certainly I, but many others as well, had ever encountered. And so we normally deploy the net from right here, which was not an option when it looked like this. And so unfortunately, that did really cut back the number of samples that we were able to collect on this cruise. But when the seas were calmer than this, we were still able to collect a decent number of samples from the mock nests. And this is what we caught. So on the x-axis here are just different tows from one of the two ships. So we had a bigger mock nest, another ship had a smaller one. And on the y-axis are the percent of fish belonging to these different species or groups of fish. So in green, for example, are myctophids, favorite twilight zone fish, and there's all these different species and groups. And the other two really abundant families are belonging to this bristlemouth family and also these hatchet fish. Okay, so that gives us an idea of which fish are there, and that helps us use, in some cases, more precise parameters for those species. And then what this net also told us was how much are these fish migrating? So these myctophids are migrating from the very sea surface sometimes down to about 500 meters. The hatchet fish do a slightly smaller dial vertical migration from about 100 to 500 meters. Then the bristlemouths typically don't do that dial vertical migration. They stay down deep. Okay, so we know who's there, we know where they're moving. Now we need to figure out how many of them there are. And so we do that with biomass estimation from the net catch, where we haul the nets back on deck. We measure the fish that are caught in the net. That measuring happens back in the lab. And then we can use that biomass estimate for all these different groups from all these different nets to estimate fish carbon transport. Again, what we really want to know is how much are fish transporting compared to the total passive plus active carbon transport in the biological carbon pump. And so that calculation gives us percent contribution of fish. All right, so here's what we found. That spot in the middle shows the average estimate for fish, and that line on either side is the uncertainty range. So fish transported about this much per milligrams of carbon per meter squared per day. Zooplankton, quite a bit more in this specific time and place. And then the passive sinking particle flux from dying sinking phytoplankton is, and other particles is really quite variable throughout these 3 different time periods of a 6-week cruise. So we can see in the last time period, in these last 2 weeks of the cruise, this spiked up really high, and this was measured by sediment traps that sit in the water column, catching whatever falls down. And so what this means is that, for one, fish contributed relatively modestly at this specific time and place compared to the total biological carbon pump. But there could be really high seasonal variation in this contribution of fish. We were out there during the middle of the North Atlantic spring bloom, and towards the end of the cruise, that was towards the end of the spring bloom when some of the stuff had gotten to those sediment traps at 200 meters. And so that means that, if the phytoplankton bloom is doing this throughout the year, and the contribution by fish is pretty consistent throughout the year, because these fish live several years, that means that the contribution from fish at the time we were out there might have been this. But as the sinking particle flux goes down, the contribution by fish could be a much bigger relative component of the biological carbon pump. So yeah, it's important to take these findings in the context that they were collected. So we've been talking about carbon transport past 200 meters, but that is definitely not the same as carbon sequestration. For carbon to be sequestered for long time scales, it needs to either get, quite deep. And the amount of carbon that is transported to 200 meters is much greater than the amount that actually gets sequestered if it makes it that deep. So we looked at that using this global model of ocean circulation, and we're trying to figure out, if carbon gets to 200 meters, how long is it stored? And what we found from that is that anywhere from about 8 to 30% of the carbon that is transported that deep is actually sequestered for, say, about 100 years, which is a pretty typical time scale to use when you're going to talk about carbon sequestration in terms of a climate change benefit. So Similar to chapter 1, we found these, similarly important parameters at the individual fish level from this Monte Carlo analysis we did again in chapter 2. We also found that biomass alone contributed about as much uncertainty as all those other physiological parameters combined that we saw before. And one of the biomass parameters that contributes a ton of uncertainty is this net capture efficiency. So this is when you go to catch fish in this net, how many are you going to assume are evading the net before you have a chance to measure them? And for this net, we don't know if we're really catching only 5% of what's there or more like 50% of the volume of water that those nets filter. And so this is the number one, biomass is the number one contributor to uncertainty here when we look at the ecosystem level. All right, so how did fish stack up compared to other parts of the biological carbon pump? When we propagate all this uncertainty from their physiology, from biomass, what we find is that fish contribute anywhere from only about 0.5% to the biological carbon pump to maybe as much as 18%. This is basically, fish are either not very important or super important to the biological carbon pump. So this is also like a 10, more than an order of magnitude range between the minimum and maximum estimate. And that's even quite a bit more uncertain than that global review paper we're making out, uncertainty in fish carbon transport to be. And so I think, by taking the time to propagate all this uncertainty, we got a frustratingly high but more realistic idea of the uncertainty here. Like I mentioned, if you're going to talk about carbon sequestration, you really need to convert from transport to sequestration, and whatever number is correct is the true number between this range. No more than about 1/3 of that is actually sequestered for, say, 100 years at this specific part of the ocean. All right, so we've gotten an understanding, at least from the snapshot in time, and the contribution of fish. We definitely know that there's high uncertainty there. We've identified some of the most influential parameters. So now the next, and then we've identified that difference between carbon transport and carbon sequestration, because the fish drop off the carbon at some depth, but some of that sinks deeper. There's some added uncertainty there. So taking stock of this uncertainty, I wanted to see if there was a way that we could actually reduce that uncertainty with new or better measurements on some of these influential parameters. So I mentioned biomass is kind of the number one contributor to uncertainty. Some of the ways that we could refine that is through better measurements using net systems that, for example, have cameras mounted to the front where you can actually see how many fish are evading the net. Maybe use some AI system to count the fish and identify them. That's sort of a new technology that could improve our net-based sampling of biomass. There's also kind of new or different measurements that use a combination of acoustic methods and also net sampling to get more refined acoustic-based biomass estimates. So, acoustics, you're sending down a sound wave, you're getting the bounce back of sound. And from that, you can get some estimate of fish biomass, but right now it's still pretty hard to convert from the sound that you get back to some biomass estimate. And so there are entire research programs working on these questions to constrain biomass uncertainty. So that's not something that I could, pick up as a chapter as a PhD student to do on my own. What I could do, what seemed more reasonable, is to actually measure these respiration rates and maybe get a more refined estimate of mesopelagic fish respiration rate from this work. So that's what I did in chapter 3. And going back to this figure, this is just, again, those fish respiration rate measurements that we have from the literature where there's still quite high uncertainty in how you're going to predict respiration rate from the mass of the fish and other temperature of the water as well, you need to know. And what I was hoping is that I would do these measurements and I would get these nice little points right around a model fit line and constrain some of that uncertainty by measuring fish of just the species we care about, just in the time that we care about on that 2021 cruise that I mentioned. So I tried to measure respiration rates in 2 different ways that were recommended in that big global review paper. on fish carbon transport. The first is using direct respirometry. I know some of you in the audience have done this and know what these experiments look like. You're basically measuring live animals in a chamber, and you measure the drawdown of oxygen over time. You convert that to carbon dioxide, and you can figure out how much the fish is respiring in carbon dioxide. And a disadvantage of this method is that you're measuring live animals. So yes, you're measuring it directly, but unfortunately, it's really hard to collect these fish live. They live offshore, they're expensive to get to, and they're really delicate. They don't do well coming into contact with a hard surface like this. It's not what they're adapted to. And so to get around some of these challenges, we also did this proxy measurement that was recommended as a key field study in that fish carbon cycling review paper. And in this proxy measurement, you measure fish that are euthanized, brought back to the lab, and were previously frozen. You basically take a sample of their tissue, and you do a series of different lab protocols. And what that tells you is the rate of some chemical reaction that can be assumed to be the maximum respiratory capacity of that fish. So this is, the most that fish could possibly be respiring you get from this proxy measurement. The disadvantage of this method is that, like I said, it's a proxy. So you then have to be able to convert from this measurement of maximum respiratory capacity to a realistic measurement from a live animal. And I thought, no problem, I'll just do these two measurements with the same exact fish for both, same exact zooplankton for both. And then we'll get that conversion factor from the proxy to the actual respiration, more realistic respiration rate. So same crews, we hauled in the nets. Most of that went into biomass estimation, but where possible, this is my old boss and collaborator, Joel, helping me sift through the catch and find whatever live animals we could that were still in good enough shape to do these respirometry measurements. So we'd collect those fish, we'd hurry them down into the lab, and then I would set up these respirometry experiments in a dark room. We can't perfectly simulate the natural environment in a lab, but we did our best. And we only used, Myctophids were the only fish that we used for this. And those were collected from the very last net to come up out of the water that was sampling the sea surface. So they weren't in the net for very long. They weren't undergoing a big pressure change by the time we measured them. So, so what you do is you put the fish in this chamber, and you measure the drawdown of oxygen over time. You refresh the water, you measure it again, and so you get multiple respiration rate measurements per individual animal. Yeah, we did this for Myctophids. We didn't manage to collect any other live fish families on this cruise. So we also did it for these two really abundant groups of zooplankton that go into, that were going to be used for that zooplankton carbon transport estimate. So that's what I was doing on our ship. Meanwhile, my collaborator, Amy Maas, was out on the other, one of the other ships and doing all these respiration rate measurements on live zooplankton that were of these different groups. So these are some of the smaller zooplankton that were really common at this study site, including salps and different copepods and pteropods. And so that's the respirometry work. The proxy measurement, like I said, you go into the lab, use previously frozen fish. You can do this on as many fish as you brought back from sea that you want to do it for. So we got a much higher sample size from this. And so I measured this proxy measurement in these 5 different really abundant taxa that made up a big biomass of our catch. Meanwhile, Amy was planning to do it for those smaller zooplankton to really capture, all the diversity of the catch in these 2 different respiration rate methods. But tragically, Amy's online. I hope this doesn't make her too sad to remember this day, but Amy's samples melted in the process of shipping. And so we weren't able to do this method with her samples. So we were left just with these guys. But, these were still, the Myctophids in particular, where we did the respirometry and the proxy measurement, that was valuable because that conversion factor had never been measured in mesopelagic fish before. So, you do these several months of lab work, we get the proxy measurement for about 100 different individuals. And then that's it. That's the end of the data collection. So what did that look like? This is the respirometry work, again, with the live animals. And what we found was the data is the data that we collected in our study, and the red are all those literature values from that IKETA 2016 paper on fish, and also this other IKETA 2014 paper that synthesized everything we know about zooplankton respiration rates. And what we found was that, unfortunately, I didn't get as tight of a relationship as I was hoping for, but we end up with about the same amount of uncertainty, whether we use our estimates alone or just the literature estimates. But what we did find that was interesting was that we have higher than predicted values, about 4 times higher on average respiration rates than what we would have assumed if we had only been basing out on the literature values. So this was kind of surprising, and a lot of people just have to, because they're not using direct measurements. They just have to assume this literature-based estimate. And so it was still useful to do this and to see that at this specific time and place where there's really high food availability, the metabolic rates of these animals may have been higher. We ended up with quite a bit higher prediction of respiration rate than we would have if we had used only the literature estimate. All right, what about that proxy measurement? Maybe that's how we're gonna nail this uncertainty. What we found was that what we were hoping to find was that these proxy measurements would all be right around a really specific conversion factor, and that we would know exactly what that conversion factor is for fish versus different types of zooplankton. But what we found instead was that we had this really big range in what that conversion factor was between different individuals. So yeah, on the x-axis is the conversion factor, on the y-axis is how many individual fish or zooplankton had that value. And so what this means is that when you have the conversion, the proxy measurement, we're not sure exactly how to convert that to the direct respirometry because it varies a lot. So you can't just go and do this on fish that don't have the direct respirometry and know exactly what that respiration rate would have been. All right, so chapter 3, in summary, showed us that we had ended up with a pretty low sample size from this direct respirometry. There was still relatively high variation, similar to what we get from the literature. And so that didn't really constrain this respiration rate uncertainty much. Using this proxy measurement, we had a much higher sample size, which was great, but we had also really high variation because of uncertainty in that conversion factor. Something to know about that conversion factor is that people often assume in studies on fish and zooplankton carbon transport that value is 0.5. And so what we found was that might not be a super reliable assumption in converting that proxy measurement to respiration rate. So from this, I sort of concluded that this uncertainty seems to be here to stay, and that at least by doing this particular method of sampling, we weren't really able to constrain that uncertainty further. So where does this leave us? We know that fish contribute to the biological carbon pump. And in certain times of year, in certain parts of the ocean, that could be quite a high contribution, but it's really not possible right now to estimate how much. And as we saw, there was really high uncertainty range, even when we did super detailed measurements at a specific study site. But I think there's an opportunity here to use the parameters that we identified as really influential to prioritize which measurements would give us the most kind of bang for our buck in constraining uncertainty in the future. So where do we go from here? We know that there's interest in using these fish as fisheries. We also know that these fish play an important role in food webs. They are known to be eaten by tunas and swordfish, marine mammals, some seabirds that forage when they come up to the sea surface at night. And this is a project that I'm kind of working on the side to try to quantify the importance of these fish in food webs. And we know they play some role in carbon cycling, but it's really hard to estimate exactly how much. So what do we do with all this uncertainty from a decision-making standpoint? For fisheries managers, they're going to have to make a decision at some point in balancing trade-offs between using this new untapped resource for food versus leaving these fish in the ocean for these other benefits that they provide. And, it doesn't have to be all one or the other. And so I think there's a range of options here that people could choose to take. On one hand, kind of at one extreme, you maybe open up mesopelagic fisheries with very little constraints, and we might not know the impacts that will have on these other benefits. On the other end of the spectrum, you might completely close mesopelagic fish to future exploitation and potentially, lose an opportunity to meet the growing demand for food. There's also kind of an intermediate set of options where maybe you open small experimental fisheries and you really carefully monitor the impacts on these other benefits of these fish to see, what is the trade-off there between harvesting or leaving them. I also think it's really, I'm really interested in the implications of this work for carbon management. So from a climate change mitigation standpoint, decision makers kind of have two different types of options for how to mitigate climate change. We can focus on reducing our emissions in the first place. So this is kind of turning down the faucet of emissions into the atmosphere of carbon dioxide and other greenhouse gases. Or you can focus your efforts on making the lands or the oceans absorb and store more carbon dioxide than they are right now. And I would say there are definitely some exciting opportunities here for strategizing to capture more carbon from the atmosphere. But I'm definitely a big fan of the first option of, before all else, reducing our emissions in the first place. And I would say for ocean-related carbon dioxide removal strategies, altering how we fish is just not a super reliable way to get an actual climate benefit in the end. And so I would say, again, there's more reliable options out there for mitigating climate change that we know are going to have a more direct impact on our atmospheric carbon dioxide concentration. So to sum up, I learned that the uncertainties in the biological carbon pump in general, but especially in the contribution of fish, is really large. However, we can prioritize by really focusing on the parameters that matter most and constraining this uncertainty. I think creative problem solving and new technologies and ways of measuring these different things like biomass can get us closer to a more precise estimate down the road. And in the end, I think it's really important that we as scientists communicate honestly about what we do and don't know so that decision makers can make the most informed decisions that they can in how we manage our natural resources and how we choose to strategize around climate change mitigation. All right, so thank you so much, everyone, for listening. I want to go through some acknowledgements. I don't have enough time to thank all the people that really deserve thanks, but I'll start with my committee and my advisors. Thank you so much, Tim and Ray, for everything you've taught me and trained me on in becoming a scientist. And I'm fortunate to have had really wonderful role models in my advisors and in my committee members. I also really appreciate that Tim and Ray have fostered really positive lab environments and communities, and I've really benefited so much from that sense of community in my two labs. A huge thank you to Joel, who got me interested in this topic in the first place when I was working in his lab before starting grad school, and for all your help at sea. Amy Maas was texting me in the middle of the night trying to figure out these respirometry experiments when she was doing hers on her ship and I was doing mine, and I really appreciate all your support, Amy, on the physiological and bioenergetics type science that was incorporated into this work. And thank you, Jodi, for bringing a little oceanography perspective to this work as well, and for being my GSR. I also want to thank my funding sources, the SAFS Fellowship, and also the GRFP from NSF. I definitely owe a huge thanks to these crews that were out on the ships that we used data from. This was the Sarmiento, the ship I was on, and this was the James Cook, the other ship that was collecting zooplankton data. None of that work would have been possible without these huge contributions from others as well, that was measuring, the zooplankton and passive sinking particle fluxes. And finally, thank you so much to my lab mates, my classmates in this grad program, the other faculty members who I've had an opportunity to take courses with and work with. I want to give a special shout out to the staff grad community, which I think is a really unique and special part of this particular department at University of Washington. I've, gone through some highs and some lows in doing this dissertation, and without you all and family and friends support, I wouldn't be here finishing it. And yeah, special thanks as well to Nolan, who has seen me through those highs and lows and and supported me every day of the way in getting this dissertation completed. So that's all I have, and with that, I'd love to take questions. From online folks or in person, we'll make sure we give online people a chance as well. Sorry, I wasn't even. Well, I'll let Morgan moderate the online questions, but yeah, Dad.
Flux zone calculation, then, if it's a shallower sea, that might only, be less than 100 meters deep. Same thing is happening in a much shallower column of water. Has there been any studies done on shallower seas?
Yeah, for sure. So, sorry, I'm just going to close this for a second. Thank you, online people. So sweet. So yeah, there's been quite a bit of work on these coastal or ocean-based nature climate, they call them nature-based climate solutions. For example, in more shallow systems, mangroves, seagrasses, other types of systems are known to absorb some amount and store some amount of carbon dioxide from the atmosphere. The thing is, in shallower water, there's less room for carbon to get very deep where it's stored on very long time scales. So unless that carbon is getting into the sediment, for example, in a really shallow coastal zone where it's not mixing anymore with the water, you're going to get mixing from those shallow waters up to the sea surface on the order of a few years. And so that's not providing as big of a climate benefit as if you can get carbon down to, say, 500 or 1,000 meters where it could be stored for decades or centuries. So there definitely is work on that because, again, that's a part of the carbon cycle that our actions can more directly impact, right? These coastal zones that are our infrastructure, our food systems, our recreation are more directly impacting. But yeah, I think with that, again, it's really important to think about not just, for example, how much is a coastal kelp forest sequestering in a given year, but how long is that carbon being stored? And is it really being stored for, say, 100 years? For some of our local kelp species, Miranda, I've learned a lot about this from Miranda here, they die back every year. So that's not, storing carbon for a long amount of time. Yeah. Yeah, Chelsea.
Thank you, Helena, for that beautiful presentation. That was fantastic.
Thank you.
You'll be shocked to learn that I have a question.
Oh my gosh, that is so surprising.
Myctophids and other mesopelagic fishes get all kinds of cool multicellular parasites and also pathogens, viruses, bacteria, protozoa. How do you think infectious disease plays into these carbon budgets, and do we have a hope of measuring that?
That's such a cool question. Yeah, thanks for asking that, Chelsea. So for online people, in case you couldn't hear, Chelsea said you will be surprised to hear that I have a question about parasites. Chelsea is our resident parasitologist in our department, so not that surprising. And she asked a good question about whether it's possible, and have we thought about how parasites could actually be impacting the role that these fish play in carbon cycling. And I can definitely say, I haven't looked at bacterial or other microscopic pathogens or parasites in these fish, but when I was working back in Joel's lab and at Hui doing all these dissections of mesopelagic fish from our catches, I would often find these anisakid worms, I think they're called, that were all over the tissues. Yeah, I remember learning from you, Chelsea, that you know, some of these parasites actually impact the behavior of these fish, where, for example, if a parasite, let's say a parasite needs to go from a myctophid into a red-legged kittiwake. These are one of the birds that forage at night and actually amazingly consume myctophids when they're up at the sea surface. If that parasite needs to go from a fish to a bird, it may be advantageous for the parasite to cause that fish to spend some time up at the sea surface, maybe during the day when it can be caught more easily, or just you know, coming up to the very, very surface where they're easier to catch. I'm totally speculating here, but if that's the case, if parasites are impacting the movement ecology of these fish and how long they're spending at different depths, for example, then I do think it's possible that parasites could play some role in these carbon cycling questions, and I do not know what that role is, what that impact is, but I think that's a cool opportunity for future research that hasn't really been done yet. Morning.
A question from online from Colleen Patrick, who says excellent pieces, Helena.
Thanks, Colleen. Yeah, thanks for asking that, Colleen. And yeah, thanks for being here. We measured respiration rates for the fish in the at the temperature that was at the sea surface of that study site that we were all out there. Colleen was involved in that cruise as well. So the fish were measured just at that one temperature. We were using flow through water from the ship, so it's just a little bit warmer than the temperature of the sea surface by the time it got to the lab. And we did that only at one temperature. For the amphipods, I basically had a little bit of extra time at the end of the cruise to measure the amphipod respiration rate at 2 different temperatures. So we do have that for amphipods, so we can get at that like Q10 value. Yeah, Trevor.
Yeah, I was just wondering, so obviously the fish are distributed mostly sort of close to the coast where there's lots of phytoplankton production, I assume. But also the carbon we're producing. That means that the carbon uptake in the ocean from these biological processes kind of matches where it's being produced, or is it so the carbon kind of spread equally across the whole atmosphere and the fish are not?
Yeah, that's a great question. I hadn't thought about that, like, closer loop between how our emissions and our coastal systems are taking up carbon. I don't know the answer to that. I think, you know, that's a really a kind of atmospheric question in a way of how quickly is that are those emissions distributing homogeneously throughout the atmosphere in coastal systems versus offshore. But I think, well, one thing I can say is that these fish actually do live quite offshore as well as in coastal zones. They kind of, I guess, fill a niche that in areas where there aren't, you know, sardines and anchovies and other of those really abundant coastal pelagic fish, they're still out there in those deeper, farther offshore regions. So I don't know the answer to that, but it'd be interesting to know, you know, what is the time scale between carbon being emitted and carbon actually getting into some of these animals and stored deep. I don't know that work has really been done yet.
Well, I think we've reached the end of the public portion of the defense. Let's thank Helena one more time. Committee members in the room, you stay here. Committee members online, also please stay where you are. How about we plan to like reconvene like 10 minutes meeting? People can't.
That sounds great.
Okay.
Okay.
That's it.
Thank you. Thanks everyone for coming. All right. Thanks for being here.